All right, so we're here today just following up with our SARE Grant and evaluating how wine cap can improve soil health. To recap, 2 years ago at the start of the study, we created 15 plots. 5 bare plots or control plots, 5 mulch-only plots, and 5 plots planted with the wine cap mushroom. This way we can measure plant and soil health across variable ground conditions. We then test the plants in the plots for the uptake of the antioxidant ergothioneine we're interested in. So what we discovered with tomatoes is that the fruits are very juicy, so they're more challenging to test for the ergothioneine, and tomatoes themselves don't take up as much ergothioneine as some other vegetables like black beans. So what we're going to do today is we're going to plant the black beans with rhizobium into the soil mix, and then we're also going to have 2 different treatments, one with Arbuscular Mycorrhizal Fungi and one without. Recent research has indicated that it is the AMF fungi, or that Arbuscular Mycorrhizal Fungi, that actually does the transporting of the ergothioneine from the soil up into the plant. That was a lot of information, so let me break it down a bit. First, let's talk about rhizobia. So the first step today is actually treating these black bean seeds with the rhizobium inoculant. So these rhizobium will help the black beans with the nitrogen fixation. And this is important because the soils we are working with the wine cap research haven't had black bean or other crops like this grown in them for some time, if ever. So this inoculum will help just eliminate that extra variable and help the plants themselves with nitrogen fixation. What is AMF fungi, do you ask? It is Arbuscular Mycorrhizal Fungi, which is a specific type of mycorrhizal fungi that should be in the soil that can actually inhabit the interior tissue of some plants, including black beans. And recent research indicates that it's the AMF fungi that actually do a lot of the transporting of materials from the soil outside to inside the plant cells. So we're going to have 2 rows of plants in each of the plots, one treated with AMF fungi and the other with a mock inoculum without the AMF fungi. We're going to actually mix each of those into separate parts of soil and then plant those seeds into that treated soil. All of the seeds we work today will be treated with the rhizobia. For planting our beans, we're inoculating our soil with a ratio of 10% AMF to soil by weight. The potting soil mix that we're using is the Pro Mix BX, and this mix is specifically used because it has no mycorrhizal fungi or fertilizers that could skew the results of our work. We start by hydrating the potting soil to the same moisture level as the AMF inoculant. After the potting soil is properly hydrated, we measure out the proper amount of AMF inoculant and mock inoculant and mix them into separate batches. Using these mixes, we fill our seed starting trays, labeling them either non-AMF or AMF. Next, we move on to black bean seeds. All right, so now we're going to inoculate the black bean seeds with the rhizobia bacteria. And this package is enough for 8 pounds of black bean seeds, and we only have about 1 1/4 here. So we're only going to use a small portion of this. Add a little bit of water just to moisten them and mix them really well so they're kind of coated with the rhizobia inoculant. After this, we finally get to planting our seeds. Today we're starting about 700 black bean plants. Again, we have 15 plots and we need 2 rows of approximately 20 plants in each row for each plot. We're also doing a few extra just in case some of the seeds don't germinate. All right, so now we have our over 800 black bean seeds planted in both of those treatments, and we're going to keep them under the grow lights over here. Hopefully the seeds germinate in the next several days, and we'll keep an eye on them for healthy growth. And then once they're ready to be transplanted, we'll be placing them or planting them into the plots outside.